engulfed by a cell, they can antagonize or resist being killed. And so the adaptive immune system, again, can address those disease-causing characteristics of the pathogen. So a helper T cell, an effector helper T cell, on one hand, can provide activation signals that would induce these macrophages to consume oxygen and generate free radicals, superoxide ion, as well as hydrogen peroxide. Those are very bactericidal, and that would ensure the killing of bacteria that have resisted the basic mechanisms of bacterial killing by macrophages. And so this is held under tight control and requires, in most cases, an adaptive immune response, because this also can damage healthy tissue and contribute to some of the pathology of infection. So therefore, it's held under tight control in that macrophages that have engulfed bacteria that are antagonizing, that are have evolved characteristics to avoid being killed by the macrophage, the adaptive immune system can override those characteristics and address that problem by providing additional activation signals that would activate an additional type of killing that macrophages possess. But again, it's held under tight control because this can cause tissue pathology. Some of these reactive oxygen intermediates and reactive nitrogen intermediates can damage host cells as well as the microorganisms that are generated to kill. And then finally, helper T cells can also provide additional activation signals for the humoral arm of the adaptive immune system. And the humoral arm of the adaptive immune system is generated to address those pathogens that are extracellular, as well as the type of products they produce that can cause disease. And so helper T cells can also provide the additional activation signals such that only the bone marrow-derived or B lymphocytes that have specificity for antigens displayed or elaborated by the particular pathogen that's causing the infection will become activated. So again, this specificity is associated with the recognition of infected cells and the recognition of cells that are participating in the same infection. And this avoids bystander activation. Only those valuable lymphocytes, only those cells that are participating or have specificity for the pathogen at hand will receive the additional activation signals to either kill the pathogen that's engulfed or to synthesize and secrete its antigen receptor as a soluble protein known as an antibody. And so that constitutes the next level of the next arm of the humoral immune system is the production of specific antibodies. So B cells see a different form of the antigen than T cells see. B cells have an antigen receptor that's referred to as an immunoglobulin. The B cell antigen receptor recognizes the native form of an antigen, the native three-dimensional structure of the antigen as it is displayed by the pathogen. Because once a naive B cell is going to be activated to proliferate and then differentiate into the effector cells of the humoral immune response, which will be plasma cells, that essentially are just antibody-secreting factories at this point, a naive B cell will recognize antigens displayed on the pathogens through its immunoglobulin antigen receptor. And once the appropriate activation event takes place and collaboration with the appropriate helper T cells that are responding to the same pathogen, the B cell will proliferate and then differentiate into effector cells that will secrete the original antigen receptor, now termed an antibody, because it's directed against another body or directed against the pathogen. And so the terminology is kind of confusing. These genes are immunoglobulin genes that are constructed in the bone marrow to give each B cell its own specific antigen specificity that will recognize the native form of the antigen as it is displayed on the surface of the pathogen, because once it secretes its original antigen receptor, that will be carried in the bloodstream back to the site of infection and specifically target the pathogen for elimination. And so let's just finish up here by looking at the job of an antibody. So this is the humoral arm of the adaptive immune system. And so it's generated to recognize those pathogens that replicate extracellularly as well as their products. So an antibody protein is an immunoglobulin protein is consists of two identical heavy chains, so-called because of their molecular weight, and two identical light chains that pair to create a bivalent antibody, a bivalent protein with two identical antigen binding sites that can recognize a particular antigen that's elaborated by the pathogen. And this, once secreted by a plasma cell as an antibody, this original antigen receptor will be secreted as a soluble protein by plasma cells that have been activated in the secondary lymphoid tissues. That antibody, for example, would leave the lymph nodes in the lymphatics, which would eventually be carried back in the bloodstream. And because we have a large number of plasma cells that are present due to the infection that day, and that's due because those few B cells out of the repertoire have an antigen receptor that has specificity for some of the antigens elaborated by the pathogen will become activated and then will collaborate with helper T cells responding to the same pathogen to receive additional signals to proliferate and then differentiate into effector cells. And so effector cells, their effector function is to secrete the original antigen receptor, which will be like a specific bullet that will target the pathogen that will be carried in the bloodstream, back leak into the site of infection. Because the site of infection, there's an inflammatory response taking place, plasma proteins are leaking in, eventually a high concentration of these antibody molecules will leak into the site and then will bind to the pathogen, will bind to the same antigen again, which is elaborated on the surface of the pathogen or its products, and therefore determine how the pathogen or facilitate specific removal of pathogen. And so this essentially shows how antibodies work. So they're produced, there's bacteria in the extracellular space as well as their toxins, and in some cases this could also be viruses, for example. So this could be small molecules, this could be toxins or viruses, as well as the whole pathogen. And so the antibody will recognize just a small specific feature of that pathogen, an antigen. And therefore the antibody will bind to the surface of that pathogen, and that makes the recognition and elimination of that pathogen by commandeering the cells of the innate immune system, that makes the recognition and elimination of that pathogen much more efficient, such that the infection can now be cleared. So in this particular case, the adaptive immune system can overcome certain characteristics of this microorganism that antagonize phagocytosis by macrophages and neutrophils. Because macrophages and neutrophils, they have receptors not only for activated complement proteins on the surface of microorganisms, they also have a receptor for the C-terminal or constant conserved regions of an antibody protein. So that makes recognition and elimination much more efficient. Because some pathogens can mutate around some of the broadly specific phagocytic type receptors and antagonize being recognized and eliminated, making elimination much more inefficient. And that's why an infection was established in the first place. So the adaptive immune system can address these particular problems posed by pathogens that allow them to establish an infection. And then finally, it's not shown on this slide, but in both cases, both for T lymphocytes and B lymphocytes, once activated, memory cells will form that will have the original antigen receptor with the original pathogen specificity. And so the memory cells will be long-lived cells that, due to most natural infections, will persist for the life of the animal. And so now, specificity for a particular pathogen that causes an infection. Initially, if it's a new pathogen that the animal has never encountered before, the repertoire of lymphocytes that can recognize it is on the order of 1 in 10,000 to 1 in 100,000. However, memory cells that develop, that expand in number, it's on the order of about 1 in 100 to 1 in 1,000. So the next time this animal encounters a pathogen, lymphocyte activation will occur in just a couple, about 3 days, instead of about 7 to 10 days, because it takes, time has to be taken for antigen to be delivered to a secondary lymphoid tissue and for populations of lymphocytes to enter that secondary lymphoid tissue, and then those few lymphocytes that can recognize the pathogen provided by the antigen-presenting cell will then have to enter the cells, be induced to enter the cell cycle, proliferate, and then differentiate to effector functions. Finally, the adaptive immune response can address the infection. But memory cells will be activated much more readily because characteristics of the pathogen and their effector function will be imprinted already on those cells. So they will enter the cell cycle much more rapidly and then proliferate and then immediately display their effector function and prevent the pathogen from causing disease a second time around. So it'll be a much faster and a much more efficient response. So if a pathogen causes an infection, the adaptive immune system can adapt to that particular pathogen and prevent disease from a natural infection a second time around. Are there any questions?
Let's thank Todd.
Okay.